We can go ahead and get started. Welcome to another week of Solanaceae seminar online. Today I'm introducing to you Jia He. So Jia He received his undergrad degree in School of Life Science at Peking University, China, before coming to the United States for grad school. During his PhD, Jia worked with Scott Cawthon's lab studying the developmental functions of microRNAs and vegetative phase changes and quantitative relationships between microRNAs and their target genes. Jia developed a deep interest in development, genetics, and evolution. He then joined the Lippmann Lab at Cold Spring Harbor Lab in 2019 as a postdoc researcher and has been focusing on the genetics, genomics, and development biology of Physalis, an emerging Solanaceae reference system. So with that, you can go ahead and start sharing your screen, Jia.
Yep. Wonderful. Thank you very much for the opportunity and for the introduction. So is everybody seeing the slides okay? I'll start to play it. Let's see. It's good?
Yep.
Okay, great. Okay, so hello everyone. My name is Jia. I'm a postdoc in Zach's lab at Cold Spring Harbor Lab, and today my title is Establishing two Physalis genomes as new Solanaceae systems enables genetic re-evaluation of the Inflated Calyx Syndrome. And since I'm the first one from the Lippmann Lab to present in this seminar series, I'll have a very brief introduction of the lab. And the Lippmann Lab here, we are located at the beautiful campus of Cold Spring Harbor Lab. And you can see the lab is in this historic building, called Delbrook building, right at the harbor. So if you look outside of the window, you can see the beautiful view of the harbor. And if you're coming to the lab early or work hard till late, you'll be rewarded by this beautiful burning clouds of the sky. And the Lippmann Lab primarily uses tomato as a reference system to study the genetics related to floral development. So in the summer, we spend a lot of time in the field, and throughout the year, we also have some nice greenhouse space to grow our plants. And a majority part of the effort of the lab focuses on a fundamental biological question, which is the relationship between genotype and phenotype in an evolutionary context. So without going into details of many of the research, I would say that over the years, people in the lab made some quite interesting discoveries that led us to think more about some basic concepts in genetics, such as epistasis, cryptic genetic variation in evolution, structural variant versus trait variation in domestication, and gene dosage and compensation, and etc. And people also develop genetic tools to engineer quantitative traits and apply that to crop improvement. And throughout the years, we developed wonderful collaborations to several groups of scientists, highlighting here today, and you will hear more about in today's talk, is the collaboration of our lab to Joy's lab at BTI. So several years ago, a former postdoc in the lab, Zach Lemon, started to use this orphan crop, Physalis, also known as groundcherry, for his study. So Physalis is very similar to the wild tomato, hasn't been through much of domestication, so it shares a lot of characteristics of wild tomato, such as the indeterminate growth and small fruits and etc. So by applying our understanding of the biology of floral development and domestication in tomato, Zach Lemon used CRISPR to knock out homologs of the tomato genes that we know to be important in domestication traits. So when he knocks out cloud 1, he obtained a larger fruit with a little bit more local number. And by knocking out cell pruning, he obtained this determinant compact plants. And by knocking out SP5G, another important gene in the floral pathway, he can create this moderate determinant plants with denser fruits along the shoot. So this was the first case that the lab uses Physalis as the genetic system. And going beyond that, beyond using it as a test bench of our knowledge, Physalis also helps us to track the evolution of the compensation relationship between cloud3 and its parallel. So this is the recent work by Tak and his colleagues, and I'm very excited about this work. So I'll go over it very briefly. As we know that shoot meristem is maintained by the feedback loop between cloud3 and wushu. And cloud3 is a signal peptide which has a close related paralog in tomato called Cl9. When the signal peptides are lost, wushu is unchecked, so usually we'll have this overproliferation of meristem. However, this Cl9 gene is not present in all Solanaceae species. In some species, such as eggplant or Benthamiana, there is no Cl9. While in some other Solanaceae species, such as groundcherry, Cl9 is present. So there is a very interesting compensation of Cl9 going on with cloud3, that in tomato, in cloud3 mutant, we have this moderate increase of branching and fasciation. While in a double mutant, cloud3 and Cl9, we have this very dramatic overproliferation of meristem, creating this branching and extreme fasciation phenotype. This is because that Cl9 is a moderate compensator for loss of cloud3, so it can partially compensate for cloud3 loss. However, in Benthamiana, because there's no Cl9, so the loss of function of cloud3 mutations will create this very similar branching phenotype as the double mutant in tomato. It's interesting, in groundcherry, where there is presence of Cl9, the single mutation of cloud3 has pretty much very little phenotype when compared to wild type, while the double mutant cloud3 Cl9 has this very similar phenotype of branching and overproliferation of meristem as in tomato. So this means that Cl9 is a strong compensator in groundcherry. So it can compensate for most part of the loss of cloud3. And Tak then went on to unravel the molecular mechanisms underlying this strong compensation. And this is very interesting to me, and it also highlights the need to have multiple genetically trackable systems beyond what we have or what we primarily work on to use as a reference. And more than that, there is another extraordinary evolutionary novelty that is called Inflated Calyx Syndrome. Here I'm referring to as ICS in Physalis. As you can see here on the left side, we have this nice, healthy Physalis grisea plants growing in the greenhouse conditions. And after anthesis, we can observe the dramatic increase in size of the sepal, primarily at the lower part, the part below the sepal tips, to create this balloon-like structure to encapsulate the fruits. And if we peel open the calyx, we can see that there is ample space between the fruit and the calyx. In order for us to kind of appreciate this dramatic phenotype here. I have a clip of a time-lapse video of the calyx inflation. If you watch the flower here at the 6 o'clock position, you can see it start the bottom, the base of the calyx start to inflate at a very dynamic way. And within a couple of days, we can see dramatic increase of the size. And within, several hours of time, when we see this inflation, we can already visualize that increase in the sepal size. So we can assume that the molecular event leading to this will be a very dynamic event. So historically, people were interested in the molecular mechanisms underlying ICS, and the first attempt to understand the molecular mechanism of ICS was in 2005, and the researchers were prompted by some observations in other species. For example, when the matchbox16 gene in potato is overexpressed, we usually observe this increased sepal size as shown in the left graph. And there's another matchbox gene called MAT11, which is closely related to MAT16. And when the homologs of MAT11 from different species such as maize, tomato, or Arabidopsis is overexpressed in Arabidopsis, people observed increased sepal size. So they found the homologs of those two genes in Physalis and named them MPF1 and MPF2. And they used RNAi to knock down the expression of those two genes. And they found that in some lines of the MPF2 RNAi, there is reduced leaf size and reduced calyx size, which are highly correlated with male fertility of these lines. So they also overexpressed the MPF2 in potato, and they observed increased sepal sizes. So they concluded that it is the heterotopic expression of MPF2 to be critical to the evolution of ICS, and MPF2 is a kind of master regulator of this process. However, there are several points that I want to mention of these experiments, such as that in some of their lines, although, for example, if you look at their lines on the graph on the right corner, you have this knockout line that shows similar degree of reduction in calyx size, but the MPF2 expression level is pretty similar to wild type. So there's a little bit of concern in these areas. And because they observed the correlation of male fertility and calyx inflation, they went down to study fertility and hormones that's related to fertilization and the relationship between these to the calyx inflation. So they did a bunch of ablation studies to ablate stigma or fruits at different stages. And they observed that if you ablate the stigma at the early stage, you can stop the calyx from developing. And in many cases, if you apply exogenous cytokinin or GA, you can partially rescue this inflation. So they formed this model that MPF2 is linked to fertilization, and cytokinin gibberellin are fertilization, pollination, the fruit-related hormones that are incorporated into this calyx inflation pathway. And later on, the same group of researchers found that an AP1 protein, MPF3, also to be involved in this pathway. So what they did was to use RNAi and VIX to knock down MPF3. And here on this middle panel, panel E, they observed that the tip of the sepal in the MPF3 knockdown lines are much larger than that of wild type. However, in the fully expanded calyx, I think the MPF3 knockdown line has very similar calyx inflation as the wild type. The size is a little bit smaller, but the calyx inflation looks pretty similar. And because they also did some molecular experiment to study the interaction between MPF3 and MPF2 and formulate that MPF3 is repressing MPF2. In their genetics experiment, it's a little bit confusing to me that when they combine MPF2 and MPF3, both two knockdowns, double knockdown lines, they observe this quite normal and comparable to wild type inflation. While in these lines, the MPF2 expression level are very similar to the MPF2 level in the MPF2 single knockdown lines. So with the same MPF2 expression level, you are having distinct phenotypes of the calyx inflation, which is a bit confusing to me. And also, this group of researchers, they did some phylogenetic analysis of the evolution of ICS that gave them this very surprising result that if you look at the graph on the left, all those bold red lines represent the Physalis species, the species with MPF2 expression in the calyx and with the inflation, while the species with the blue boxes are the species with MPF2 expression but no calyx inflation. So this means that MPF2 is not sufficient to cause calyx inflation. At least there will be other factors functioning in this pathway. And it's also surprising to the authors that, as I quote here, given the complexity of the requirement for ICS formation, it's surprising that this trait appears to have arisen several times independently in the Solanaceae and even within the Physalis, as suggested by our phylogenetic analysis. And there's a more recent work from Stacy Smith lab that Grisea look at the evolution of this morphological novelty that she characterized this calyx simulation into three categories. One is the non-crescent calyx, which is that the sepals are very small and smaller than 50% of the fruit. And the second category is the crescent pressed calyx, which means the calyx are a little bit bigger than 50% of the fruit, but not encapsulating the whole fruit. And the third category is the inflated calyx, which means the calyx fully surround the fruit and with space between the fruit and the sepal. And through the phylogenetic analysis, she found that the evolution of calyx inflation usually conforms from non-crescent to a crescent pressed and to inflation, and there's a very rare reversion of this evolution trajectory. And inflated calyx has been evolved independently multiple times. So there's a bit of a contradiction to the mechanism of the evolution of the ICS. So combined with the previous reasoning that we need a new system, I think it's time for us to have a very good genome of Physalis to help our understanding of this inflated calyx and also some other important aspects of Physalis. So we started to build reference genomes of two closely related Physalis species, the Physalis grisea and pruinosa. These two species are very closely related to each other, and they're intercrossable, and they differ in several minor phenotypes such as leaf shape, leaf size, and pigmentation patterns. So this work is a collaboration for our lab to the Mike Shan's lab. In Mike's lab, we have Mike Lange and Sri to help with the assembly of the genome and annotation. So basically, we used PacBio and ONT combined with Hi-C sequencing to build the chromosomal level assemblies of those two Physalis species. And we used RNA-seq reads to build the annotation and also lift over the annotation from other Solanaceae species. And in the end, we have two chromosomal level genomes, which are very good quality. We have about 1.4 gig size of each genome, and each genome has a chromosomal level. And as we can see here, those two genomes are very close to each other. We don't see a lot of differences between these two, yet there are what we call structural variations. We can see that if we call structural variant using pruinosa reads compared to grisea, we have a distribution of deletion and insertion of SVs at moderate size. And many of those SVs intersect with gene features that may give us functional consequences of the gene product. So after we get those two genomes, the first thing we look at was to demonstrate the utility of these two genomes, that we decided to look at the pigmentation pattern differences between those two species. As you can see in this picture, in grisea, we have this purple brownish nectar guide, while in pruinosa, we have the yellow color of this guide. And this pigmentation difference is also observed in the stems of those plants, that in grisea, we have this purple stripes along the stems. So we used the F2 population of this, the cross between those two species, and we used only a small number of plants, like 20 plants of the yellow pool and 20 plants of the purple pool, to do this rough bulk segregated analysis. And as we can see here, we have a strong signal on chromosome 4 that the majority of the chromosome 4 are above the 95% cutoff, confidence interval cutoff. So this means that this gene, the causative genetic variation controlling this color pattern, is on chromosome 4, but because of the low recombination rate or the low number of the plants we use, we cannot proceed to pinpoint the gene at this point. But luckily, we know that these mutations are highly likely to be related to anthocyanin pathway, and people in historically used petunia to basically laid out this nice anthocyanin synthesis and regulation pathway, as abbreviated here, that a lot of mutations were isolated in petunia that created ranges of color patterns in petals. And notably, many of these regulation parts were on these late biosynthetic genes of anthocyanin. And there's the MBW complex, which is consists of those MIB and HLH, and WD40 transcription factors. They have important roles in regulating these late biosynthetic genes. And on the right, I'm showing here is different alleles of mutations in anthocyanin 1 or anthocyanin 11 that can create different color patterns. So we look at all the genes and see which ones are located on chromosome 4. And we have only 4 genes on chromosome 4 and only 1 gene that shows a genetic variation among those 2 populations. So we have the anthocyanin 1 gene that has a 43 base pair deletion in the second intron from the pruinosa, which is the yellow guide pool. So as you can see here from the map, all mapped reads, and also from the assembled genome, that there is a 43 base pair deletion in pruinosa. Then I went to check the molecular phenotype, molecular consequence of this deletion. And by using RT-PCR, we can clearly see that in pruinosa, as well as the F2 yellow bunk population, the transcripts are longer than that of the grisea, which is the purple form. And after we sequenced the amplicons, we found out it's because of the 43 base pair deletion, we have a intron 2 partial retention, which causes the transcript in the pruinosa to be much longer than that of grisea. And within that included intron, we have a premature stop codon, which made the transcript inactive. So of course, we CRISPRed the anthocyanin 1 gene in grisea, and in the T0 chimeric plants, we already started to observe this loss of anthocyanin pigmentation in stems and in flowers. So a brief summary of this first part is that we generated high-quality chromosomal level genomes of grisea and pruinosa. And using the genome annotation, we mapped the pigmentation pattern phenotype to be caused by the genetic variation in the anthocyanin 1 gene, resulting in the loss of the N1 function. So after we demonstrated the utility of these 2 genomes, we then turn into the much needed genetic reevaluation of the inflated calyx. So we generate CRISPR lines of MPF2 and MPF3. So we have multiple loss of function alleles of MPF2 and also multiple loss of function alleles of MPF3. And surprisingly, that we didn't observe any calyx inflation related phenotypes of these plants. As you can see here, the CRISPR mutations of MPF2 and MPF3, they have this intact inflation process. And in MPF3, we observe similar phenotypes from their previous knockdown, RNAi knockdown, or VIX line, that we have this increase of the sepal tip. But the base of the sepal, where the inflation was happening, does not change much. Of course, you can see some shape changes to the calyx, but the inflation is intact. And there's a short video about wild type versus on the left versus MPF2 of their inflation, and it seems that the inflation is intact and not affected at all. And of course, we observe some other phenotypes of the MPF3. One of such is the increase of branching. As you know that the sympodial growth unit of Physalis usually contains one leaf, one flower, and 2 branches in the wild type. However, in MPF3, it's frequently we'll see 3 branches. And we observe some changes in calyx height over width ratio, so that the MPF3 calyx appears to be a little fatter. But as I mentioned, the overall the inflation program is not affected. So we started to think, okay, can we get some of the calyx phenotype from some other candidates that showed that their orthologs in other species show some sepal-related phenotypes. So the first set of candidates were the tomato agamas-like 1 gene. So the TAGL1 gene in tomato, why we think it has some function in calyx development was that there was a mutation we identified to show this pseudo or similar increased calyx phenotype in tomato, and this is caused by transposon insertion in front of this TAGL1 gene that causes it to have higher expression. So with this overexpression of TAGL1, we also observe this increased sepal phenotype in tomato. And TAGL1 has another close related gene called TAG1, tomato agamas 1, that there's also evidence that when overexpressed, TAG1 will create some sepal inflate-like phenotypes. So we CRISPR out the 2 genes, and as you can see here, because these 2 genes are involved in the C-type function of floral development, so we observe homeotic transformations of stamen to these petal-like structures. And we can see this, the calyx size are a little bit smaller than the wild type. However, the inflation program is intact. You can see even for this small inflated calyx, it's much larger than the calyx from the flowering stage. So the calyx inflation is still there. And another set of candidate genes are the SEP4 genes that in tomato, we show that the mutations in EJ2 and LEN will create enlarged sepals. So we wonder if we can see calyx phenotype in Physalis if we knock out these genes. Again, when we use CRISPR to knock out LEN, REN, and EJ2 in Physalis, we observe no disruption of the inflated calyx. In EJ2, we observe some phenotypes of the sepal shape, very similar to the macrocalyx, that the tip of the sepal are turned more leaf-like and more elongated. But overall, if you look at the base or the majority part where the inflation is happening, you don't see a dramatic phenotype. And because previously I mentioned that people propose that fertilization is an integral part of ICS. Also wonder if we mutate many of the AP3 type genes, which have function in floral organ identity, we would disrupt the fertilization pathway, and in this way, will we be able to create some calyx inflation defects? So we CRISPR out TM6 and the GLO1 and the AP3, which is DEF. And again, even when we see the shape or the geometry of the calyx changing, and even when we see that the petal is turned into another layer of sepal, the sepal inflation is still present. So this linked us back to the question of the role of fertilization in calyx inflation. As we can see here, this is the previous TAG1 mutant I show. We can see that even without inflation and without the fruit within the calyx inflation program is still intact. And if we look closer to this set of mutants, TAG1, TAGL1, DEL, and GLO1, we can see that we have various degree of disruption of the floral organs. And there's no self-fertilization of this because they either lack the stamen pollen or they have defective carpels and stamens altogether. So all those lines, they show the inflation of calyx, which means that although hormone or fertilization may feed into this calyx inflation pathway, they're not absolutely required. So there has to be an innate pathway of calyx inflation. So a short summary about this part is that we obtained CRISPR mutations of MK2 and MP3, and they did not disrupt ICS. And despite various degree of floral phenotypes, the CRISPR mutations of other candidate genes did not disrupt ICS either. And mutations that disrupt floral organ and self-pollination didn't demonstrate that fertilization is not required for ICS. So at this point, we thought, okay, maybe we want to try some forward genetics to screen for mutants that can have some functions in calyx development. So I did a small-scale EMS mutagenesis in Physalis grisea and planted them out during the pandemic in the Riverhead field. And I was lucky to find a mutant that has no husk when I look at it in the field. So I call it huskless. So if we look at the phenotype of this mutant, it's very interesting that it seems that the first word of this huskless has a shared role, a shared identity of sepal and petal that at an early stage start with greenage leaf-like structure, but also having this pollen nectar guides that's only found in petals, and it only has 3 words of this mutant. So as the plant progress into the fruiting stage, the petal, this whorl, start to go through the same senescence program as normal petals do. So there's a naked calyx in the mature fruit stage. So here's an example of what you will observe if we look at the plants when they bear many fruits. So because we have a very nice genome and good annotation, we also, before that, I'll show you a couple SEM images to see that in normal developing calyxes, we have 4 whorls, which is the carpel, stamen, petal, and sepal. However, in this mutant, only 3 whorls are observed at very early stages. So because we have very good genome and good annotation, I started to just use direct sequencing to map this mutation. So I sequenced the mutant in the field and its wild type looking siblings, and to try to filter for mutations that are homozygous in the mutant, but not homozygous in the wild type pool. And after the filtering and analyzing the potential function of these variants, there are only 8 high impact genes, and one of them is an AP2-like gene. And as we know that AP2s have function in floral organ editing, so this is my top candidate. So I did a co-segregation test of this G2A SNP in this AP2-like gene. And this A genotype completely co-segregates with the huskless phenotype. So I went to look at the molecular consequence of this G2A change by amplifying the transcript of a mutant versus wild type. It's clear that because of this G2A change, this AG spike site is moved inside this exon, causing a partial skipping in exon 5. So CRISPR of this mutation shows, here I'm showing the mutation of the CRISPR, which is 122 base pair deletion in the first exon and part of the UTR. So in this mutant, we see a very similar phenotype to that of our EMS mutant. So if we look at this gene and of its homologs in other species, it's interesting that in Petunia, actually this gene has been studied and called Ben. And more interestingly is that in Petunia, there's another closely related paralog of Ben called Bob. And this paralog is present in tomato, but absent in Physalis. So in Physalis, we only have this huskless, but no paralog of huskless, such as Petunia. And from people study in Petunia, we know that Bob and Ben are redundantly required for proper floral development. As you can see that in a single mutant of Ben, the petal development is pretty intact, and we only observe the tip of the sepal to be petaloid. However, in the double mutant of Ben and Bob, the development of petal is highly disrupted, and in many cases, there's no petal, and people will observe partial conversion of sepal into petal-like structures. So this is pretty similar to what we observe in Physalis, although they have some minor differences in terms of phenotype. But this is another demonstration that because of the gene duplication or the paralogs, that we can have different phenotypes if we knock out one gene in certain species. Because I observed this shared identity of this first wall, so I wanted to see if it is really a shared identity of sepal versus petal. So we sequenced transcriptomes of these first wall of huskless and compared that to the sepal wall or the petal wall wild type. So as you can see from this PCA analysis, that the first wall of huskless is always having a transcriptome profile in between the wild type sepal or the wild type petal. So it has this shared identity of those two walls. And it's very intriguing to me that this shared identity is kind of in early stage, it has more sepal identity, while in later stage, it has more petal identity. So to wrap up this findings, so we use forward genetics approach to identify huskless mutation in an AP2-like transcription factor that eliminates inflated calyx by merging the first and second word as a single word of shared sepal petal identity. And in summary, our resources and findings meant Physalis has a valuable new addition to Solanaceae genetic systems and force a reset in the search for the mechanism underlying the evolution of ICS. And also, these resources open the door to study many other Physalis traits of fundamental interest, for example, like secondary metabolism, disease resistance, or some of the like sugar content, which can help to accelerate improvement of this novel berry fruit. So with that, I would like to thank people that are involved in this work. So first, I would like to thank people in the Lippmann lab, Zach Lippmann, as a wonderful mentor and role model for us young scientists, and people in the lab, including postdocs and graduate students and technicians, to create this wonderful scientific atmosphere. And I would like to highlight Joy's lab that contributed significantly to our CRISPR of all these mutations. And Michelangelo Ree from Mike Schatz's lab for the bioinformatics of the genome assembly and annotation. And also other plant group members in Coachman Harbor. And also the founding sources, Coachman Harbor, HHMI, and NSF. That's my talk today, and I'm happy to take any questions.
Thanks so much, Jia. That was great, super comprehensive. Definitely a huge fan of your research. If people have questions, they can either type it in the chat or you could raise your hand. I think Stacy has a question, so you can go ahead and unmute Stacy.
Can you hear me? I can't see
Yes.
myself, but thanks for that. I am like, poof, I am blown away by that huskless mutant. I had a couple questions. So actually first about the, it was AN1, right? The AN1 mutation.
Yes.
In the first part. So you did that. I took notes, hopefully I get it the right way. So the pigmented one is grisea, and you did like a CRISPR knockdown of AN1 in grisea, right? To recover
Yes.
the pruinosa phenotype. Did you all consider going the other direction and trying to like overexpress grisea's AN1 in pruinosa to show that its overexpression or expressional line, not over, just regular, like just expression of a functional AN1 would produce a pigmented phenotype in pruinosa? Because that seems to me to be a stronger test of the role.
Yeah, so we haven't done that. Yeah, but that's a good point. Yeah. So so one thing is that for this study, it was primarily to use as a test case of that we can rapidly, map these genetic variations. But you have a very good point that, if you want to really, study the functions or evolution of these genes, and we could use that, express the wild type or the functional version of N1 in pruinosa. And yeah, it's interesting that as you know that there are many other species of Physalis that also show this yellow polygon phenotype. So it's interesting also to see if we can find similar variation in those species, or if there is some other polymorphism that contribute to this yellow phenotype.
Yeah, well, partially it's this maybe me being personal interest. I don't know. I have a proposal where we would do try at least some transient overexpression in flowers across species. And it would be nice if that was worked out well in Physalis, because then it would be easier for us to do an iochroma too. But I also had a question about the huskless. That just is so wild. Like what a totally wild mutant phenotype. And I wondered, why do you think it affects whorl number in addition to the identity of the whorls? Like, why not make two sort of tepaloid whorls instead of just like, why the reduction in whorls in addition to the transformation of their identity into this tepaloid kind of thing?
Yeah, I think it's still, I mean, honestly, I don't know the answer, but it is I think for example, it has to do with many of our understanding of the floral development. That's our understanding of this, the key functions of these genes that are coming from a few species. As for example, as you see that they're closely related genes in Arabidopsis is actually not the tall genes, which are not involved in floral identity, rather in floral timing. And when people study these genes in Petunia, they notice that there's like functional diversification happening. And in Petunia, it's a different story, a little bit different story, because there's redundancy of those two genes. And in Physalis, because of the absence of this parallel, I can capture, this function, this mutation in the mutant screen by only take one gene to capture this phenotype. So I think there's still a lot unknown to be unraveled, and it will be interesting down the road. But right now, I don't have the answer.
Yeah, no, I was very curious. I mean, as you were speaking, I went to Google to try and remember sort of which floral identity genes were fairly confident of their function in Solanaceae and how broadly they do the same thing. So like, do we know all the A, B, C, E class genes in Solanaceae and what their jobs are and how well conserved is that across the family? And it seems like maybe it's not, maybe that too is a bit of a challenge. But
right. Yeah. Even when people studied some of these functions in tomato, this seems to be a little bit different. And there's another layer of regulation of these genes, that these genes are regulated by microRNAs. So I can envision this. So there's another layer of like age-related layer into the regulation of that. For example, in some of my mutants, I can observe that in later, the older plants of the huskless mutant, you can have smaller sepals that kind of like the oppressor crescent phenotype in Rossia's definition. So there's like definitely other layers to this.
yeah, it's fascinating. My last question, I'm sorry, I had so many questions, was in terms of trying to think if there could be, if huskless, or genes that are, part of the same developmental network are involved in other taxa. I wondered if for these other convergent origins, and maybe Rosia or Chelsea know too, in those cases, is it also true that the development of the inflated calyx happens with or without fertilization? Because for some reason I thought in some of those other taxa, maybe, it wouldn't be triggered unless the flower was fertilized. So it seems like that kind of comparative study would be really useful as a first pass, but I don't know if that's known or not outside of Physalis, or even if it's for sure like conserved across all of Physalis. Okay, Rosia has a question. Maybe she's going to tell me, or maybe Jia.
Yes, maybe we can hear about Rosia's question for you.
Yes, as far as I know, there is not much information about if fertilization is needed in other taxa. Like the best study is Physalis, and in that previous study, they were saying that fertilization was needed, but now we know that it's not. So I don't know actually if there is, at least probably in Kenji there is some work, but at least I think that fertilization is not needed in Kenji.
I know that some
greenhouse.
Yeah. So in the greenhouse, I don't think it's super common, but there's definitely like a Hedrophilia species that if it's not fertilized, it will produce a calyx, but it's definitely reduced than what it would be if it was actually fertilized. So there's something going on there that it still produces its calyx, but I would say most of the time, like Physalis, it will just die. It's not going to produce its inflated calyx. So I think most of the time it won't produce a inflated calyx if it's not fertilized, but I think there's those rare occasions where it will occur, but I think they're definitely reduced in size. when I've seen it.
Yeah. So what I view this is that when you have this like innate pathway of this inflated calyx, but we know that these hormone signals generated by either fertilization or fruit development, these hormones are also part of the apparatus to for like cell division, cell expansion. So I think it's definitely, it makes sense that this can feed into this pathway. So to facilitate or, make facilitate the inflation. So I can definitely, understand that part. So it's not to say that fertilization has nothing to do with the inflation. Is that I think the key point is that there is like an innate pathway of the calyx inflation.
Right. And my question actually was about the MPF 2 and 3. And as far as I recall, you have tried to have mutants of these genes separately. Have you tried to have a mutant of both genes to see if there is some disruption in inflated calyx?
Yeah. So I'm in the process of having a double mutant. But I mean, the thing is that according to previous interactions of these genes, yeah. So my take is that because of the single genes, they are showing no phenotype of this process, and because of their interaction, the proposed interaction, personally I wouldn't expect that that interaction to produce a significant result, but I'm in the process of making that double mutant. Yeah.
Yeah. And I have actually a second question that it's about that huskless phenotype that is amazing. And I was wondering, like the calyx seems to not be present even in the bud. So do you think there is some disadvantage in the development of the flower there because there is no protection at all? That is the main function of the calyx.
You mean in the huskless mutant?
Yes, the huskless.
Yeah. So yeah, I know that we kind of assume that there's the function of the calyx will be, either to increase the photosynthesis or protection of the fruit. But for my observation, in a huskless mutant, because of the mutation, you have probably like affected the fruit set overall or the yield overall. And as I mentioned before, it has a little bit, age-related factor into that. So in a lot of those early flowers, sometimes you see the flowers or the fruits to abort. And in this case, you won't have fruits. But as the plants grow a little bit bigger and older, you can have these naked fruit phenotypes very consistently.
Okay, thanks.
All right, Stacey has another question.
Sure.
Okay, it all went so fast for me. Okay, so so you did, I'm trying to remember. So did you do a CRISPR knockdown of huskless, like to recreate the phenotype, or is that something that?
Yes, I did CRISPR of the huskless,
Okay.
and it shows a very similar phenotype.
Okay, I can kind of see that in slide 49. What I was wondering was, did you sequence the transcriptome of that? Like, I was just thinking what would be really interesting to help understand how this is working and what it's doing is to see like what other genes were downregulated when you knock out huskless or upregulated.
Yeah. Yes, so I have that. I have the comparison of transcriptome, but I haven't just really the deeper analysis of like which genes are upregulated versus downregulated. So because when doing this, we kind of have to consider that you compare this huskless, this whorl versus either the petal or the sepal of the wild type, right? You can only compare to one of them. You cannot just compare to a combination of these two, because you don't know like the different gene expression of the contribution of their expression in sepal versus petal. So yeah, so I do have the RNA-seq data set of huskless versus either wild type petal or wild type sepal, but haven't dived deep into that to see which genes are differentially expressed.
Yeah, that's a good point. That's a complicating factor. What to compare it to. So cool. Thank you.
Thank you for the questions.
So I have one question too. So you talked about the chromosome 4 having low recombination rate. Do you think it's a super gene? And I know you mentioned that there was like 4 different anthocyanin genes on there, but do you know what other genes are within that block?
Yeah, so right now I don't have the other genes top of my head, but I checked those whether there are genetic differences of those genes, and I didn't see much of those genes. The only polymorphism I observed was in anthocyanin 1.
It might be interesting to see if it's the S locus that's on that chromosome, because that tends to be a super gene technically, and I actually don't know where it is in Physalis, and that might be a reason why there's low recombination, which would be kind of interesting. But
okay,
yeah, cool. All right, other questions, other comments? Cool, thanks so much, John.
Thank
It was
you.
great. You've done so much work. It's so comprehensive. Excited to see where you go from here. So join us in 2 weeks. We're going to be talking about a potential new potato species with Rodrigo and Nicolo. So see you then. All right, bye guys.
See you.